Hello class, our today's topic is transgenic animals. Transgenic animals may be defined as animals that have their DNA manipulated to possess and express a foreign gene and are known as a transgenic animals. The genome of their animals have been changed and they can carry the genes from the other species. Example of this transgenic animals includes the rats, rabbits, pigs, sheep, cow, monkeys, and fish. Although over 95% transgenic animals are mice. This is the picture of Dolly. Dolly was the sheep that was cloned by the Roslin Institute of the Edinburgh, Scotland, the first transgenic sheep to produce alpha 1 antitrypsin was the Tracy. But the question is how these transgenic animals produce. What are the methods used for the protection of these transgenic animals? The first method is the DNA microinjection with the help of DNA microinjection. Second is with the help of the retrovirus mediated gene transfer. And third is the embryonic stem cell mediated gene transfer. So the name Dolly, Polly, and Molly. You are familiar with the Dolly, but the Polly and Molly that born in 1997, the two Eves, were the first mammals to have been successfully cloned from an adult somatic cell and to be transgenic animals at the same time. This is not to be confused with the Dolly the sheep, which was the first animal that mammal to be successfully cloned from an adult somatic cell, whereas there was no genetic modifications carried out of the adult donor nucleus. The transgenic monkey, that name is ANDI, was the first genetically modified monkey, and the green fluorescent protein is found in that monkeys was inserted in this monkey's chromosomes. The two main regions are very common main regions that why are these transgenic animals being produced? Why we produce these transgenic animals? The two most common reasons are that some transgenic animals are produced for the specific economic trade, for the investigation purpose, for the discovery purpose, and other transgenic animals are produced as disease models, or we can say the animals genetically manipulated to exhibit disease symptoms so that the effective treatment can be studied. And the benefit from these transgenic animals we can study broadly under three heads, that in the field of the medicine, in the field of the agriculture, and in the field of the industry. In the field of medicines, we develop these transgenic animals like cows that produce the human protein enriched milk, and in the field of agriculture, the larger sheep that grow more wool, and in the field of industries, the goat produces that is spider silk for the material production in the field of industry. In the field of the medicine, the region to produce the transgenic animals are very common and very useful, and it is for the five near about purpose reasons. One is the normal physiology and development. So these transgenic animals are designed to study the different aspect of the genes, like how genes are regulated, how they are going to affect the normal function of the body and its development. For example, that is study of the insulin-like growth factor that involved in the growth can be done by introducing genes from other species that alter the formation of this factor and study the effect that result. Also information is also obtained from the biological role of the factor in the body. So for the normal physiological development, we produce the transgenic animals. Second reason is to study the disease. This is very important. The transgenic animals models for the human disease, in which investigations of new treatment for any diseases, and also gives understanding how genes contribute to development of that particular disease. So transgenic animals models uh, is used in the study of the disease like cancer, cystic fibrosis, rheumatic arthritis, and Alzheimer disease also. Third reason is that in the field of the biological products, the products produced by the living organisms, some of which are medicines, are very very expensive. So transgenic animals can be created by the introducing of these genes with codes for a particular products, induce that organism to produce the desirable product in medium. For example, just like human protein alpha 1 antitrypsin. This protein is used to treat the emphysema in case of the human being. So attempts are being made for the treatment of phenylketonuria, PKU, and cystic fibrosis also. In 1997, the first transgenic cow, that name is Rosie, is produced human protein enriched milk near about 2.4 grams per liter. The human alpha lactalbumin, and it is a nutritionally more balanced product for the human babies than the natural cow milk. This is the picture of the Rosie, that transgenic cow. And next is the for the vaccine safety, there is a requirement of this transgenic animal also. For vaccine safety, vaccines are first tested on the transgenic mice that developed for use in testing laboratory before they are going to used on the human being. So transgenic mice are being used to test the safety of the polio vaccine. Was very common in India. If successful and found to be reliable, they could replace the use of the monkeys to test the safety of the batches of the vaccine. So after testing on the mice, then again testing on the monkeys, the vaccine has to be declared that it is safe for the human being. So it may be used for the trial in the human body. The next is the chemical safety testing is also required. It is used with the help of the transgenic animals. The transgenic animals introduce with the genes which make them more sensitive than the non-transgenic. So transgenic animals are more sensitive with that genes. So chemical testing may be done on the transgenic animals. They are then exposed to the toxic substance and affect the study. Toxicity testing in such animals will allow us to obtain the result in a very less time. You can find here a different animals, transgenic animals. It is created by the human being with the help of the biotechnology, and for the testing and investigation of all these steps. But in the field of the agriculture, the breeding quality and disease resistance is also maybe tested with the help of these transgenic animals, and in industrial applications also. But there are so many ethical issues, and it is also known as the bioethics. Thus, ethics includes a set of the standard by which a community regulate its behavior and decide as to which activity is legitimate and which is not. So, therefore, we can say that the bioethics may be viewed as a set of standard that may be used to regulate our activity in relation to the biological world. So the manipulation of living organism by human race require certain regulations. These ethical standards are required to evaluate the mortality, morality of all human activities. So it is our morality that might help to or harm any living organisms. So it is the ethical issues. We should not harm the living organism for our benefit. Genetically modifications of organisms have unpredictable. We don't know what type of organism is to be introduced into the ecosystem. So result, when such organisms are introduced into the ecosystems, it may be destroy, it may be damaged, it may be create problem in our ecosystem. Indian government has set up a organization that name is the GEAC. The full form of the GEAC is the Genetic Engineering Approval Committee, which will make the decision regarding the validity of the GM researches, genetically modified researches, and the safety of the introducing GM organisms for the public services. The modifications that uses of living organisms for public services as food and medicine sources, for example, has also created problems with the patients also. So use of the genetic material, plants and biological resources by the companies that granted with the patent has triggered public anger since they have been identified, developed and used by the farmers and peoples of a specific country. Over 2 lakhs variety of rice that is found in India, which make India richest in the field of the rice. So basmati rice, one type of rice which unique aroma. You know that it have a very good and unique aroma and flavor, and they are near about 27 documented variety of basmati that is grown in the India. In 1997, an American company got patent this right on basmati rice through the US Patent and the Trademark Office. This will allow the company to sell a new variety of basmati in the US and abroad. This new variety of basmati has actually been derived by the crossing with the semi-dwarf variety and claimed as an invention or a novelty. The patent extends to functional equivalent, implying that other people selling basmati rice could be restricted by the patent. So there is some protocol of this patency. So these are the controversies regarding the patency and biopiracy. Several attempts have also been made to patent uses, products and processes based on Indian traditions, that's herbal medicine, turmeric and neem also. We should be vigilant and counter these patent applications, else other countries or individuals may encash on our rich legacy and we may not be able to do anything about it. So we have to watchful for all these things. One thing that is biopiracy. Actually, biopiracy is the use of the bioresources by multinational companies. So the patenting of the plant genes and other biological products that are indigenous, it is found in any country, it should not be go without any patency to another country. If it is goes, that is known as the biopiracy. Use of these bioresources by the multinational companies and other organizations without proper authorization. So if it is not authorized from the countries and the people concerned without the compensatory payment, it is known as the biopiracy. So developed countries patent the knowledge and resources of these underdeveloped countries and enjoy the immense profit. And the industrialized nations are rich financially but poor in biodiversity and traditionally knowledge. So, in contrast, the developing and underdeveloped world is rich in biodiversity and traditionally knowledge related to the bioresources. So, traditional knowledge related to bioresources can be exploited to the develop modern applications and can also be used to the same time, effort and expenditure during their commercialization. This realization of the justice, inadequate compensation, and benefits sharing between the developed and developing countries that led some nations to develop law to prevent such unauthorized exploitation of the bioresources and the traditional knowledge from one country to another country. The Indian Parliament has cleared that the second amendment of the Indian Patent Bill that takes such issues into the consideration, including the patent terms, emergency provisions, and research and development initiatives. So, this is all about the this chapter number 12. Thank you and have a nice day.